So our third of the macromolecules is the protein. Proteins are made up of subunits called amino acids. And amino acids are fairly simple molecules and not complex, not too complex. They're very small. And we begin to associate amino acids together to form proteins. So proteins are made up of a series of amino acids. When we combine one amino acid with another, we do so by forming a special type of covalent bond called a peptide bond. So when we add one amino acid to a second amino acid and they bond together in dehydration synthesis, we'll call that a dipeptide. But a little more on that here in just a sec. I want to get to the basic structure of an amino acid. And the basic structure is surprisingly similar among all of the 20 different types of amino acids found in the human body for purposes of forming a protein. You'll notice that all of these examples shown on this figure have a central carbon. We know carbons form 4 bonds, and the first bond we'll talk about that's formed from the central carbon is a hydrogen, a simple hydrogen. You'll see it below each of the carbons here on this figure. To the left of each of the carbons is an amino group, an NH2, and that's in blue. On the right side of the carbons, and the third bond coming off the carbon is a carboxyl group, a COOH. And so it seems like they're really, really similar, and they are, because the only difference is in what lies off the fourth bond of the carbon, and this is called a radical group. Radical groups, there's 20 different radical groups because there's 20 different amino acids. Radical groups can be as simple as a single atom, a hydrogen, and if that's what it is, then that amino acid is termed a glycine. It can be a chain of carbons in that fourth bond. You see that with the lysine amino acid. Or you can have a ring structure, and you see that with the tyrosine amino acid. But nonetheless, no matter how complex, you're only going to get up to maybe a double ring structure that you would see with the amino acid phenylalanine or tryptophan. That's about as complex and as large as these molecules are going to get. Now back to the formation of a protein, we have to begin at the beginning, and we're going to combine two amino acids into their simplest form called a dipeptide. And what you're going to see here is, again, dehydration synthesis at work. We have the amino side of the second amino acid bonding with the carboxyl side of the first amino acid. An OH group from the carboxyl group will combine with a hydrogen from the amino group, and you'll have water liberated. Then the carbon and nitrogen will form a bond directly together, forming what is called a dipeptide. This is a special bond, a special type of covalent bond called a peptide bond. Hence, we call the resulting product a dipeptide. And just as easily, just to point out that hydrolysis can lead to cleavage of this dipeptide. You just need to add some water, and you'll break apart that bond between the carbon and the nitrogen. So this is a dipeptide. I can keep doing this amino acid after amino acid, and I can get up to hundreds of amino acids hooked together. We call that a polypeptide. And we'll discuss what happens with polypeptides. Very unique, special features occur with polypeptides. Okay. Now, proteins can exhibit quite a variety of folding, and this gets immediately into that question: what happens when several amino acids become associated or bonded together into a polypeptide? Well, we talked about oxygen being electronegative, and we've talked about we haven't talked about nitrogen being electronegative, but it is. It's a very electronegative element. You've got some hydrogens there, you've got some electronegative elements or atoms present. Hydrogen bonds are going to form. So we're going to start to see that when we put a bunch of these amino acids and bond them together, we're going to see lots of complex folding and twisting. In fact, it occurs in 4 levels. Now, before it begins twisting and changing its shape or conformation, we're going to just talk about its alphabet. What are the sequence of amino acids that make up the polypeptide? And that's called the primary sequence. Okay. Now, once we have the primary sequence fully established, all the peptide bonds are present, then we'll begin to see hydrogen bonds forming, and we'll start to see the sequence, the straight chain of amino acids begin to fold up into unique structures. And you can see them here in part B, and they can become either a helix-like structure or kind of a sheet called a pleated sheet. And that's because of the hydrogen bond formation. Now, once you begin forming hydrogen bonds, this polypeptide, which we could also term as a protein because now it is functional. It can perform something. It can do something within the cell. But to really stabilize its features, it now will assume a tertiary shape. And tertiary, or the third level of folding, is going to result from the formation of ionic bonds. We have some sulfur groups in some of our radical groups that form off the fourth bond from the carbon of an amino acid. There's some unusual sulfurs, and sulfurs can form bridges, and these can form ionic bonds. So we'll see the establishment of ionic bonds. In a typical protein, I don't know, you might have 2, 3, 4, 5 ionic bonds formed in this way. You'll have gazillions of hydrogen bonds though. So I'm not talking a huge change, but you're adding stability to the folding of the protein with these tertiary or third level structures. Now, at this stage again, completely functional, but sometimes proteins want to associate with each other. Fully functional proteins can work to function together and interact with each other and form into what is called the quaternary structure, or the fourth level of folding. And this quaternary structure involves multiple polypeptides or proteins interacting with each other. So because they're interacting with each other, they can perform differently, perhaps activate in a different way, and you'll see a lot more when you begin discussing some of these complex proteins in ANP2. Certainly hemoglobin is what I'm thinking about right now. We'll be talking about collagen, the most common quaternary protein in the body. Sorry about my printer there. We'll also be talking about keratin, which is in the skin. And so there are a lot of these quaternary structures if we just start looking in the body. Now, we talked about at the level of activation energy needed to make a reaction occur. Remember, we have our reactants. We needed activation energy to make them interact with each other. And then that energy came down when the products to form the products. So we had to input some energy to get the products. Every chemical reaction requires some type of energy input. If it doesn't, then the activation energy is zero. It's about the best we get. But enzymes, or at the time we called them catalysts, can change this picture. We don't have to add a mountain's worth of activation energy. If we have a helper, then we can only add a much less needed energy. And so catalysts or enzymes can serve to decrease the activation energy needed to make a reaction proceed. And our type of catalyst inside the human body are enzymes. Enzymes are proteins. So this is the basic structure and function of an enzyme. It's showing you that an enzyme is a protein. It's fairly large, and it's going to act on a molecule called a substrate. Within the enzyme is a nice little site for the substrate to nestle in, to kind of get snug into. And so in this particular case, we're talking about the enzyme sucrase. Sucrase is going to work with a molecule called sucrose. We've dealt with that before. We know it's a glucose and a fructose that are combined together. So sucrose is the substrate. It will dock within this nice little site called the active site in the enzyme. And the active site then, that's where the actual separation of these two molecules will occur, and you'll get liberation of the glucose and fructose. So an enzyme greatly enhances the speed and reaction time that this can occur in. It provides a favorable environment. It orients the molecules in such a way that they are much more likely to interact with each other. Enzymes are great for that, and so they lower the amount of activation energy. So there are four things enzymes do that we need to know for the exam and just to know. You need to know that enzymes speed up reactions, or they catalyze reactions. We're saying the same thing. Catalysis equals speeding up. Enzymes are reversible. They can work both ways. Okay, so we show here the formation of sucrose, the breakdown of sucrose, but we could just as easily have shown you the formation of sucrose because enzymes commonly work in reversible conditions. All right, so enzymes are also reusable. Thousands upon thousands, if not millions of times can be used before they have to be replaced, before they're too degraded to continue being used. And fourth, they are very selective. You'll note the name of this enzyme, sucrase. That means it doesn't work with lactose. We have an enzyme called lactase that works with lactose. You have other enzymes specific for certain molecules. So it's not like they're universal and they'll work with anything to either form it or break it down, being reversible. So enzymes are very selective into what they use. So we've got speeds up the reaction, it's reversible, it's reusable, and it's highly selective. Before we stop, I want to talk about the idea that enzymes are also can be broken down or can be stopped from functioning. And there are two mechanisms involved here. One way to stop a protein from functioning is to adjust the temperature. Excessive temperature or too low a temperature can break down the peptide bonds that are found in proteins. And so proteins in the human body normally act and function between 0 to 40 degrees centigrade. Well, that's approximately 32 to 104 degrees Fahrenheit. Start to think, I think I've heard before where if I'm told by my doctor my fever gets up to 105, 106, I need to be concerned, especially when you're talking about young kids. And this is because the proteins, especially in the nervous system, are very much attuned to this temperature degradation. And what will happen is as the temperature increases, the proteins begin to wobble apart. The tertiary structure breaks down, the third level of folding. The second level of folding can stop, and then the proteins are non-functional. This process of converting from a functional state to a non-functional one is called denaturation. So temperature change is a big cause of that. The other big cause is pH. If we get too acidic or we get too basic, this is another hallmark and cause of denaturation of proteins in the nervous system as well as other parts of the body.